September 9th, 2014. In the journal Nature Communications, a team led by Columbia University's Shai Karmi and Itzik Pear, along with more than 20 researchers from Yale, Columbia, Yeshiva University's Albert Einstein College of Medicine, Memorial Sloan Kettering Cancer Center, and Hebrew University of Jerusalem, published a finding genuinely staggering in its precision. Every single Ashkenazi Jew alive today, an entire global population now numbering somewhere around 10 million people, can trace their ancestry back to a founding group of just 350 people, living roughly 600 to 800 years ago. Not 350,000, not 35,000, 350 individual human beings, whose combined descendants would go on to become one of the most genetically mapped and genetically distinctive populations on Earth. Columbia professor Itzik Pear summarized the practical, almost dizzying consequence of that finding in a single sentence that made headlines around the world. Among Ashkenazi Jews, everyone is a 30th cousin. Well, let's build out exactly how researchers arrived at a number this specific, because the methodology itself is genuinely elegant and it's worth understanding before getting to what the finding actually means. According to reporting from Live Science, the 2014 study analyzed the complete genomes of 128 Ashkenazi Jewish individuals and compared them systematically against non-Jewish European populations, specifically hunting for genetic markers unique to people of Ashkenazi descent. What they found was a genome saturated with an unusually high number of long, identical stretches of DNA shared between people who, according to any conventional family tree, shouldn't be closely related to each other at all. According to Columbia University's own research summary, by analyzing exactly how frequently these long, identical nucleotide sequences appeared among supposedly unrelated individuals in their sample, researchers were able to calculate backward, mathematically, to the exact size of the ancestral population capable of producing that specific pattern of shared genetic material. The math pointed to one number consistently across multiple independent research groups running the same basic calculation: an effective founding population of approximately 350 individuals. That word effective carries real technical weight here, and it's worth pausing on because it's easy to misread this number as literally claiming only 350 human beings existed at that specific moment in history. That's not quite what it means. An effective population size in genetics refers specifically to the number of individuals who actually contributed viable, surviving genetic lineages to all future generations, not necessarily the total head count of every person alive in that community at the time. There may well have been more than 350 actual living Ashkenazi Jews in medieval Europe during this exact period. But when geneticists trace every single genetic lineage present in today's 10 million person Ashkenazi population all the way back through history, every single thread ultimately converges on a pool of ancestors mathematically equivalent to just 350 individuals, meaning the actual surviving. Genetic contribution funneled through an extraordinarily narrow bottleneck at some specific point in the past. The timing of that bottleneck is almost as remarkable as its severity. According to World Politics' detailed breakdown of the study's methodology, the 2014 analysis pinpointed this genetic bottleneck to somewhere between 600 and 800 years ago, corresponding roughly to the 12th through 14th centuries. That's not some distant, half-mythical event buried in the earliest chapters of human prehistory. That's the same broad historical window as the Black Death, the Crusades, and a string of documented brutal massacres and expulsions targeting Jewish communities across medieval Western Europe. According to that same analysis, the demographic catastrophes sweeping through Jewish communities in Western Europe during the 12th and 13th centuries, combined with the subsequent migration of survivors eastward into Poland and the broader Pale of Settlement, created exactly the conditions capable of producing a genetic bottleneck this severe, a population violently reduced to a small number of survivors clustered in one specific location, who then expanded rapidly into a new demographic environment where conditions ultimately favored high reproductive success. And expand they did, at a rate that's genuinely difficult to fully picture. According to World Politics reporting on the study's own findings, that founding population of roughly 350 individuals subsequently expanded at an average rate of approximately 16-fold per generation, eventually producing the millions of Ashkenazi Jews alive by the era immediately preceding the Holocaust. 16-fold per generation, compounding across roughly 25 to 30 generations. That kind of sustained, explosive demographic growth, launched from such an extraordinarily narrow genetic starting point, is precisely what produced the genetic signature researchers can still read clearly in Ashkenazi genomes today, hundreds of years and dozens of generations later. There's a genuinely fascinating side discovery buried in this same research. one that directly settled a long-running historical debate about where this founding population actually came from in the first place. According to Live Sciences coverage, comparing Ashkenazi genomes against those of Flemish populations revealed that the founding group was of approximately evenly mixed Middle Eastern and European descent. That finding carries real historical weight because it directly undercuts a specific alternative theory that had circulated since the 19th century, one proposing that the core of the Ashkenazi Jewish population actually descended primarily from the Khazars, a Turkic people from the Russian steppes who had famously converted to Judaism as a ruling class centuries earlier. Study researcher Itzik Pe'er addressed that theory directly. The new genetic evidence made the Khazar origin theory considerably less likely, instead reinforcing the more mainstream historical view that Ashkenazi ancestors descended primarily from Jewish populations originating in the Levant, who subsequently settled and mixed with local European populations over the intervening centuries. According to World Politics' more detailed geographic analysis, researchers have specifically debated the exact founding location using the same genetic data, with what's called the Rhineland hypothesis, placing the founders in Jewish communities located in what is now Western Germany and northern France, proving consistent with both the historical record of medieval Jewish settlement patterns and the genetic evidence itself. A follow-up study conducted by the same broader research consortium, led by Itzik Pe'er, went even further, tracing this population's genetic history back through not just one, but three distinct historical bottleneck events, stretching across an almost unimaginably long span of time. According to Columbia University's own detailed research summary, the very first bottleneck the consortium identified occurred somewhere between 85,000 and 91,000 years ago, a period the researchers linked directly to the broader out-of-Africa migration event that produced the ancestors of every non-African population on Earth today. A second separate bottleneck occurred considerably later, around 21,000 years ago, marking the beginning of a period of interbreeding between people of European and Middle Eastern ancestry, the two ancestral populations that would eventually combine to form the specific founding group discussed in the original 2014 study. And then, finally, the third and most severe bottleneck, the medieval one already covered here, reduced the population down to fewer than 350 individuals roughly 700 years ago, the exact narrow genetic doorway through which the entire modern Ashkenazi community would eventually emerge. Now here's where the story stops being purely a matter of historical curiosity and becomes something with genuinely serious, ongoing medical and scientific consequences, playing out in hospitals and research labs around the world right now, a population that's extraordinarily interconnected, all descending from such a narrow founding group, and then, for centuries afterward, following cultural and religious traditions of marrying largely within the same community, produces a genetic profile fundamentally different from more genetically diverse outbred populations. According to a detailed research paper published examining the health implications of exactly this genetic signature, the Ashkenazi Jewish population represents what researchers describe as a genuinely unique population for genetic study, based specifically on its documented history spanning less than 1,000 years, involving a limited number of founders, at least one severe population bottleneck, and a long-standing tradition of marriage within the community, technically known as endogamy. That combination of factors carries a real double edge, one that cuts in both a genuinely useful direction for medical research and a genuinely serious direction for individual and community health risk. On the useful side, a population this genetically interconnected and well-documented becomes an extraordinarily powerful tool for exactly the kind of large-scale genetic disease mapping researchers rely on to identify which specific genetic variants actually cause which specific diseases. Relatively small, reproductively isolated populations with reduced overall genetic diversity offer real, concrete advantages for genome-wide association studies, precisely because there's less overall genetic noise scattered across the population, making it considerably easier for researchers to isolate the specific genetic signal actually responsible for a given disease or trait. On the more serious side, that same genetic narrowness means certain harmful genetic mutations that happen to be carried by even a small handful of those original 350 founders, ended up spreading through the entire subsequent population at rates considerably higher than you'd expect to find in a more genetically diverse group. According to genealogy research summarized by the Legacy Trove, this exact founder effect, combined with centuries of continued marriage within the community, explains why certain genetic conditions, including Tay-Sachs disease and specific BRCA gene mutations linked to elevated breast and ovarian cancer risk, show up considerably more frequently among Ashkenazi Jews than among the general global population. It's the direct, measurable genetic legacy of that same medieval bottleneck. Whatever rare, harmful mutations happened to exist among those specific 350 survivors got carried forward, generation after generation, into millions of their descendants, at a frequency far higher than pure random chance would ever produce in a larger, more genetically mixed founding population. There's a genuinely strange, almost paradoxical practical consequence of all this playing out right now in an area most people wouldn't expect: consumer DNA testing and genetic genealogy. According to the Legacy Trove's detailed explanation of this exact phenomenon, because nearly every modern Ashkenazi Jew descends from that same tiny medieval founding population, because centuries of subsequent endogamous marriage kept concentrating and reconcentrating those same shared genetic segments, ordinary Ashkenazi Jews taking a standard consumer DNA test today frequently discover what appear to be startlingly close genetic matches with complete strangers, matches that home DNA testing algorithms often misinterpret as evidence of a recent close family relationship, a second or third cousin, when in reality the actual family connection may be 500 years old, diluted and recombined across dozens of generations. That specific phenomenon has its own technical name, a genetics hall of mirrors created by endogamy, where cumulative small shared DNA segments from potentially hundreds of distant centuries-old relationships all stack directly on top of each other, fooling even genuinely sophisticated modern genetic matching algorithms into reading ancient, diffuse, community-wide relatedness as recent, close family kinship. And there's a genuinely moving human dimension to this same genetic phenomenon too, one that goes well beyond disease risk calculations or genealogy website confusion. According to that same reporting, this exact pattern of deep shared genetic interconnection has, in real documented cases, helped reunite family members who were separated by the Holocaust, reconnecting relatives whose paper records had been destroyed or lost entirely, purely because DNA testing provided a genetic thread that traditional documentation simply couldn't. The same underlying genetic bottleneck that creates real challenges for medical researchers trying to isolate disease-specific mutations, and real confusion for people trying to build an accurate family tree from a home DNA kit, has also occasionally functioned as a kind of a genetic archive, quietly preserving connections between people whose families were violently, deliberately torn apart within living historical memory, connections written directly into their own DNA long after the paper trail documenting those same relationships had been destroyed. So what does the Ashkenazi Jewish genetic bottleneck actually demonstrate in the broader context of human population genetics? It's a genuinely extreme, extraordinarily well-documented example of exactly what geneticists call a founder effect, a population passing through a severe demographic bottleneck, a small number of survivors, driven by real historical catastrophe, subsequently expanding rapidly, within a specific cultural and religious community that maintained strong internal marriage patterns for centuries afterward. That combination produces a population with genuinely distinctive, measurable genetic characteristics, reduced overall genetic diversity, an unusually high degree of shared ancestry among seemingly unrelated individuals, and an elevated frequency of specific disease-causing mutations that happen to be carried by the small founding group. It is not evidence of any population being genetically pure in some meaningful biological sense, and it says nothing whatsoever about superiority, inferiority, or any kind of hierarchical ranking between different human populations. It's simply a real, mathematically precise, historically grounded case study in exactly how demographic history, catastrophe, migration, and centuries of cultural marriage patterns can leave a permanent, traceable signature directly inside the DNA of an entire modern population. Millions of people strong, all ultimately descended from a group of survivors, small enough, as one researcher put it, to fit inside a single wedding reception. The genetics legacy of that medieval bottleneck isn't an abstract statistical curiosity anymore. It's a set of specific named mutations that doctors and genetic counselors work with every single day, and the actual numbers behind them make the scale of that founder effect concrete in a way the population math alone doesn't quite capture. According to research published on population-based BRCA1 or 2 testing, two specific BRCA1 mutations, known by their technical names 185del-AG and 5382-C, along with one specific BRCA2 mutation called 6174del-T, together account for a genuinely remarkable 90% of all BRCA-related mutations found in the entire Ashkenazi Jewish population. 90% concentrated in just 3 specific genetic variants, all of which trace back to that same narrow founding group. That same research found these 3 mutations combined are carried by approximately 1 in every 40 Ashkenazi Jews, a carrier rate of about 2.5%. Put that number directly beside the general population rate for comparison, and the scale of the founder effect becomes genuinely striking. According to a detailed breakdown published by Genetics, a UK-based Jewish genetic health charity, roughly 1 in 40 people of Ashkenazi Jewish origin carries a BRCA mutation, compared to just 1 in 250 people in the general population. That's a carrier rate roughly 6 times higher than the broader population baseline, concentrated overwhelmingly in just 3 specific mutations, rather than being spread across dozens or hundreds of different possible genetic variants the way BRCA mutations typically appear in more genetically diverse populations. That same source notes a further, more subtle detail worth including. Sephardi Jews, a genetically and historically distinct Jewish population with a separate migration history running through Spain and the broader Mediterranean rather than through medieval Germany and France, show a meaningfully lower carrier rate of roughly 1 in 140. Still elevated above the general population, but nowhere near as concentrated as the specifically Ashkenazi bottleneck produced. That level of concentration, 90% of all BRCA mutations, condensed into just 3 specific genetic variants, has actually reshaped how genetic testing itself gets designed and deployed for this particular population. According to that same population-based BRCA1 or 2 testing research, focusing genetic screening specifically on these 3 known founder mutations, rather than sequencing the entire BRCA1 and BRCA2 genes looking for any possible mutation, offers real practical advantages for Ashkenazi patients specifically. It's considerably cheaper, faster, and meaningfully reduces the odds of returning an ambiguous result, a so-called variant of uncertain significance that leaves both patient and doctor unsure what to actually do with the information. That same research explicitly places Ashkenazi Jews alongside 2 other well-documented founder populations, where this exact same targeted testing strategy works for the same underlying reason: Icelanders and French. Canadians, both of whom carry their own distinct, concentrated founder mutations, traceable to their own separate historical population bottlenecks. This is the detail that matters most for understanding this story accurately, and it's worth stating directly. The Ashkenazi Jewish bottleneck is a genuinely extreme, extraordinarily well-documented example of a founder effect, but it is absolutely not unique as a phenomenon. It's one instance of a pattern geneticists have identified and studied across numerous, genuinely unrelated populations scattered all over the world, each with its own separate history, its own separate genetic isolation, and its own separate set of concentrated founder mutations. According to research on BRCA founder variants, published examining populations in Jordan, the same basic effect shows up clearly in several other well-known cases. Ellis van Creveld syndrome concentrated among the Amish community in North America, cystic fibrosis showing elevated frequency among the Hutterites, and congenital chloride diarrhea appearing at unusually high rates specifically among Finnish populations. Each of these represents the exact same underlying genetic mechanism playing out in a completely different community, with a completely different history on a completely different continent, a small founding population, geographically or culturally isolated from surrounding groups, whose limited genetic starting pool happened to include a handful of specific harmful mutations that then spread through the entire descendant population at rates far higher than pure chance would predict in a larger, more genetically mixed group. Iceland offers one of the clearest, most thoroughly studied parallel examples anywhere in the world, and it's worth understanding briefly because it strips away any question of this pattern being somehow specific to Jewish populations. Iceland's population descends overwhelmingly from a relatively small number of Norse and Gaelic settlers who arrived on the island starting in the 9th century, and the country's geographic isolation, combined with centuries of limited immigration, produced exactly the same kind of genetic bottleneck and subsequent founder mutation concentration documented in the Ashkenazi case. Norwegian research examining BRCA mutation spectrums directly confirms this parallel, noting that founder mutations in BRCA1 and BRCA2 have been separately documented and extensively studied in Ashkenazi, Jewish, Polish, Norwegian, and Icelandic populations alike, each population carrying its own distinct set of founder variants shaped by its own distinct demographic history. Iceland's genetic isolation became so well documented, in fact, that it eventually spawned an entire dedicated genetics research company, Decode Genetics, built specifically around studying disease genetics within this uniquely well-mapped, uniquely isolated national population. Beyond BRCA mutations, specifically, the broader Ashkenazi Jewish genetic profile carries several other well-documented founder conditions, each with its own specific medical significance and its own real-world screening infrastructure built directly around managing it. Tay-Sachs disease, a severe, ultimately fatal neurological disorder affecting young children, became one of the earliest and most successful examples of population-wide genetic screening specifically targeting a founder mutation. Gaucher disease, a metabolic disorder affecting the body's ability to break down certain fatty substances, similarly shows up at meaningfully elevated frequency, specifically within Ashkenazi populations, again traceable to the same narrow founding bottleneck. That combination of well-documented, concentrated genetic risk has led directly to organized, community-based genetic screening programs specifically designed around this population's unique genetic profile, including programs like Dor Yeshorim, which screens prospective couples within Orthodox Jewish communities, specifically for carrier status of these same founder-linked recessive conditions before marriage, precisely because a population this genetically interconnected faces a meaningfully higher chance that two carriers of the same rare recessive mutation might otherwise unknowingly marry each other and have children affected by these serious, well-documented genetic conditions. Pull this entire picture together, the precise 350-person founding population, the medieval historical catastrophe and eastward migration that produced it, the three specific BRCA mutations accounting for 90% of hereditary cancer risk in this population, the parallel founder effects independently documented in Iceland, Finland, French Canada, and the Amish community, and organized modern screening programs built directly around managing this same genetic legacy, and you get a considerably richer, considerably more scientifically accurate story than any simple claim about racial purity could ever capture. This isn't a story about one population being genetically superior, inferior, or more authentic than any other. It's a story about how specific, documentable historical events, catastrophe, migration, and centuries of marriage patterns within a defined community leave a real, traceable, medically consequential signature directly inside a population's DNA, a pattern that shows up again and again in population after population all across the world whenever a small group